Okay, my name is Alex Scott, and this is the Unit 4 Case Connections video presentation. You can see me as you can. As you can see, my scenery has changed. I have moved. I moved last week, so it's kind of a disaster in here. I won't show you guys out there, but okay. So for the first case, I did case one. So I'll start with good evening, everybody. Today I'll be discussing the normal mechanisms of ammonia transfer in the body and how they relate to this clinical case involving a newborn girl and her suspected metabolic disorder. In the body, ammonia is a toxic byproduct of amino acid metabolism in several systems working together to manage it safely. One key enzyme is glutamate dehydrogenase, which is either the release or capture of free ammonia depending on the body's needs. When ammonia levels are high, enzymes help to trap it by favoring the formation of glutamate. Another important enzyme is glutamine synthetase, which takes glutamate and combines it with ammonia to form glutamine, which is a stable, non-toxic carrier that moves ammonia safely through the bloodstream. In tissues like the liver and kidney, an enzyme called glutaminase releases ammonia from glutamine, allowing it to be used for either urea production or to help regulate acid-base balances. In addition to these processes, transamination reactions allow the body to transfer amino groups between different molecules, concentrating nitrogen onto glutamate so it can be safely managed and again go through the body or bloodstream freely. Now, nitrogen transport is closely connected to the gluconeogenesis, especially when the body is in a fasted state. Again, from previous chapters, we know this by fasted meaning not eating. As muscles break down during fasting, amino acids are released, and alanine becomes a major carrier. Within the liver, alanine donates its amino group to form glutamate, while its carbon skeleton becomes pyruvate, which feeds into glucogenesis to produce glucose. Meanwhile, the nitrogen from glutamate enters the urea cycle to be excreted safely. So, during the fasting of the body, the body is not only making glucose to maintain energy, as we learned in previous chapters, but to also carefully disposing of the excess nitrogen within the body at the same time. In the case of the newborn, her increased urine orotic acid levels give us a strong clue about what's going wrong. Orotic acid builds up when carboamyl phosphate, which is supposed to enter the urea cycle, instead gets diverted into a pyrimidine synthase. This suggests the deficiency in the enzyme oro I always mispronounce this. I even practice this. Orinothine transcarboamylase, or OTC. I prefer OTC, much easier to pronounce. OTC normally combines carbamyl phosphate with a orthionine to create sertraline, and without it, carbamyl phosphate accumulates. This is important to note because plasma sertraline levels in the patient were normal. This helps to rule out the deficiency of the carbamyl phosphate synthetase 1, or CPS1, since CPS1 deficiencies would cause both a low sertraline and low carbamyl phosphate levels. Therefore, OTC is most likely deficient enzymes, and CPS1 is the least likely to be impaired. Finally, because the urea cycle is disrupted, arginine, which is normally produced in the cycle, becomes conditionally essential to amino acids. This means that in this patient with the OTC deficiency, arginine must be supplied through their diet in order to meet the body's needs. Arginine can just be supplemented through literally supplements from the store. I know people that take arginine supplements. Okay. For my second case, I will be doing case number 4. In this case, I'll be discussing the 58-year-old woman who presents with progressive fatigue, weakness, mild diarrhea, numbness, and balance issues. On examination, she is tachycardic, has a conjunctiva and a beefy red tongue, and a decreased sensation to light, touch, vibration in her lower extremities. Her hemocritic is low, and her megaloblastic anemia is suspected. This information, we are asked to consider which vitamin deficiency is most likely responsible. Of the choices, we have iron, vitamin B6, or vitamin B12. So, through all these possibilities, let's walk through each. First, with the iron deficiency. Iron deficiency anemia is incredibly common and typically presents with fatigue, pallor, and sometimes pekia, which usually causes microcritic anemia, meaning small red blood cells, not hemoblastic ones. Additionally, iron deficiencies would explain her neurologic symptoms such as numbness and balance issues or her beefy tongue. Next, vitamin B6 deficiency. A deficiency in vitamin B6 can cause sideroblastic anemia, which is more typically microcritic than normocritic, not megaloblastic. B6 deficiency would have caused neurologic symptoms like peripheral neuropathy, but it does not fit as well with the severe myocritic anemia or the classic glossic, which we have seen here. Finally, B12 deficiency. Vitamin B12 deficiency leads to megaloblastic anemia, glossitis, the beefy red tongue, the classically neurologic symptoms such as numbness, tingling, and loss of proprioception, which are exactly what this patient is experiencing. B12 deficiency results in impaired DNA synthesis, leading to a large immature red blood cells, and also damages the myelin in our nervous system, causing neurologic findings. So, among the three options, vitamin B12 deficiency is by far the most likely cause of her issues at the time of presentation. Now, in terms of metabolism, vitamin B12 is crucial for purine and pyrimidine synthesis. Specifically, it requires the regeneration of tachydiolfolate, or THF, which is necessary for the synthesis of thymidine, or the building block of DNA. Without enough B12, the body cannot efficiently recycle folate in its active form and bottlenecks DNA production, especially rapidly dividing cells like those in bone marrow. This is why we see megaloblastic changes. To confirm the diagnosis and rule out other possibilities, several laboratory tests can be ordered. A complete blood count should show a high mean corpuscular volume, or MCV, indicating macrocytosis. A blood smear would reveal hemosegmented neutrophils. Serum vitamin B12 levels would likely be low. Additionally, helpful tests include checking for methylmalonic acid or homocysteine levels, both of which are elevated in B12 deficiency disorders. Homocysteine alone would be elevated in folate deficiency, but methylmalonic acid is specifically elevated when B12 is lacking. Testing for intrinsic factors, antibodies, or parietal cell antibodies might also be useful, especially in pernicious anemia is suspected as an underlying cause. In total, this woman's systems and findings, or sorry, not systems, symptoms and findings are most consistent with vitamin B12 deficiency, which leads to the impaired purine and pyrimidine metabolism by blocking DNA synthesis, and the diagnosis can be confirmed with appropriate lab work. Those are the two cases that I have chosen to present. I hope you appreciate it, and I hope I did a relatively good job. And I look forward to taking the final here soon.